Thank you very much for giving me this opportunity. I really appreciate this chance, and thank you for inviting me. And I also wish to thank my colleague here, Alfred, for agreeing to join to add some depth to the aspect of the AI and remote sensing things. So with that, what is our purpose as plant scientists? And that's generating data and information that helps us to select the best genotypes, our management approaches. Sometimes that's characterizing the effect of some treatment or QTL, and to be able to detect small differences with as much accuracy as possible. So what about your opinion? What do you think is one aspect that is conducting an experiment that has the most effect on quality of the results? And I'll be sharing my thoughts on that, general principles, and that's conducting the experiment so that you get the most accurate results and make more informed decisions. And it involves creating objectives and hypotheses that define the problem clearly and suggest the desired outcome of the research. Clearly defining comparisons among treatments in a way that they can be statistically compared.
I begin now.
Use the experimental design that is best suited to the purpose, and reduce experimental error as much as possible, choosing or making experimental units that are as uniform as possible, being very careful at each step, including obtaining a perfect stand with no missing plants, and this is a challenge in Africa especially. Then analyzing the data correctly to make the best decisions, analyzing each individual environment to get the best estimates, analyzing multiple environments informatively, and understanding the contribution of each environment to the combined analysis, using a multivariate index if appropriate that often suits us as plant breeders, and a controversial one, but avoiding excessive duplicate technical analysis to reduce cost and work with little loss of overall precision. And contrary to common thinking, screenhouses and labs do not provide uniform conditions. Therefore, often RCB or even lattice is better than CRD. Here, the building on the side and the winds coming in the side affect things. The center section of this very elaborate and very good greenhouse is affected by two pathways and two walkways and the construction of the greenhouse. Now, sometimes shelves are used for experiments, and that's valid, but they also have non-uniformity and varying air temperature and movement. Research fields, too, can be very ununiform, even if it's not as obvious as the termite mounds, but other causes as well. Sprinklers, even the best ones, don't provide water in an evenly dispersed manner. Drainage ditches are important to prevent flooding, but they have an edge effect, making plots ununiform.
The slides are slow.
Oh. Okay, the path will become invisible after plowing, but the nearby test plots will be ununiform for years to come. Well, how do researchers respond to these irregularities? Avoid areas you recognize as being ununiform even if that requires a non-rectangular layout. And use spatial adjustments. Row column covariates are simple to include and help adjust for spatial patterns. And then a longer term, but very important in the longer run, is precise field maps that are geo-referenced each season, made available to future users, and also available for reanalyzing and meta-analysis. Much of the information in many previous experiments has never been extracted, yet it can be very useful. So here I just drew out a diagram of an example of a way in which the plot layout is irregular, but uniformity of experimental plots is more important than having rectangular plots. And a question often, how many reps should I use? And that depends on the expected difference among entries. Is it larger than the LSD, which can be approximated working backwards from the CV if you don't have the direct information? If expected differences are small, you'll need more reps. But if SED between entries is lowered by more reps, but the benefit of additional reps gets smaller as you continue to add reps. And if error degrees of freedom is small, more reps may be needed to have more error degrees of freedom. To see what's best, create a skeleton ANOVA and compare F critical for treatments with different numbers of reps. And what does that do for you? It gives you a smaller required F. It increases genotype mean square due to the multiplier on genotype variance component in the expected mean squares. So it increases the genotype mean squares. It reduces standard error of the mean, standard error of the difference in LSD, although that benefit decreases as I explained on the previous slide. Also, contrary to much thinking, it does not reduce the error mean square, but and standard deviation on average, but it does make it more accurate, less variable. So my master's thesis had a hybrid of sorghum and two parents to make 3 treatments. And how many reps should I have used? Well, the required F is huge if I were to use only 2 reps, and it gets lower as you go down. And at 10 reps, it's considerably lower. And so I chose to use 10 reps for my experiment, and the result turned out very good. Well, kind of shifting thought, Einstein was very keen on defining the purpose of the research. What exactly is the problem that needs to be solved? Defining that very well. But as we use various statistical methods, we need to be sure we're not torturing the data. Each method should be used in a statistically valid way. And using a log transform to make data more normal is a valid approach, but misused, it can give false results. So we need to be sure we understand the principles involved in transformations and other processes. So what about it? Are you using the most effective experimental designs? Often experiments give suboptimal decision-making power due to suboptimal data accuracy. Each step that is a leak that each step that is less than ideal is a leak. And what happens is those leaks accumulate and the quality of results goes down to where you have half the accuracy you were aiming for. So we must find and stop those leaks. And when we can reduce those leaks, reduce those inaccuracies, it really helps. Design can make a 10% difference in error mean square execution, another 10, the best analyses, another 10, giving a combined effect of 30%, which reduces the error mean square and the LSD. And although the reduction here is not huge, it's enough to be useful. And these are things that can be gained at little or no cost and effort. Now, moving on to specific designs. If we'll look at ones with fewer than 100 levels in a single environment first, and where there's less than or equal to 12 treatments, usually it's important to have 3 or even 4 reps. 2 is very iffy as far as getting power in the statistical comparisons. And in order to get the most error degrees of freedom, CRD is generally preferred unless there's non-uniformity in experimental units, which might argue for RCB. 13 to 19 plots are treatment levels. 2 to 3 reps is generally ideal with declining benefit if there's more. And the preferred design is RCB because blocking usually decreases error mean square and you have plenty of error degrees of freedom. So there's really no reason not to use RCB instead of CRD. With 20 to 200, 2 reps is usually all that's really valuable unless you really need accuracy, in which case there's 3 reps may be used. Alpha lattice is a very good one. It usually lowers the error mean square, and you can ignore the lattice blocks if they aren't beneficial, and there's really no disadvantage. The number of treatments is very flexible in alpha lattice. So I note here that above 100 is on the next slide, but actually it's after several more slides of comments. First, I wanted to compare CRD and RCB. 6 treatments, 4 reps, and CRD between divided by within gives an F critical for significance of 2.77. If we move to RCB, we lose a little bit because of error degrees of freedom, but 2.90 is only 4% less than CRD. And the impact is that RCB fails to show significance only if F-calc is between 2.77 and 2.90, and if RCB does not reduce error mean square beyond CRD error mean square. So again, use a skeleton ANOVA to decide or even to consider an advanced design. My MSc study, I wanted the 3 genotypes near each other, so that implied an RCB. How many reps? And it depends on required F and the precision of treatment comparisons. And, oops, something happened there. But my result was I used 10 reps. I decided to use 10 reps, and the result was very good. Now, many times there are two treatment factors. And if none of the factors requires handling in larger plots because of mechanics or because of cross-contamination, then that factor must be the main plot factor of a split plot design. But otherwise, both factors have few levels, and therefore you could consider treatment combinations randomized in CRD or RCB, depending on the number of reps and the number of error degrees of freedom. If there's more than 12 treatments, there's often an advantage of the split plot. And if so, the factor that has the more levels should be the main plot factor in order to get enough error degrees freedom for the main plot main plot error. And again, a skeleton ANOVA with 4 fertilizer rates, 3 rhizobium treatments, and 3 reps. If we use rhizobium as main plot, then the required F is quite high. It drops quite a bit if we use the nitrogen rate, and most of the other comparisons don't
Recording in progress.
make much difference. But sometimes the RCB is even better than split plot if there's not so many treatment combinations, and CRD can be even better than RCB. And factors in this, CRD may be better than RCB if there's few treatments, and it also depends on which factor our interaction is most critical, needs the most precision. So here I have an example of Langa Timbo's PhD research, 66 hybrids, 4 toothpick inoculations on ear rot, and there were no particular constraints. So with the main interest as the inoculation effect on hybrid was decided to be the main plot. However, there's a better, although less obvious alternative. The 66 hybrids as main plots, but randomized in an alpha lattice to better control field variability. And the take-home message from that is think outside the box and use a skeleton ANOVA to compare treatments, compare options, and consult an experienced statistician. Fisher said, asking the statistician afterward is like conducting a post-mortem. And I tell my students, I hate rescue work, trying to get a good result out of a poorly designed experiment. And I add a 4th R to the 3 R's of experimental design: randomize, replicate, restrict variability like blocking, and request help. Now, in choosing designs with more than 100 levels, alpha lattice is a good one, augmented if it's not replicated, but using plenty of checks. And P-reps or row column designs if it's not possible to have even numbers of plots of each entry. But the disadvantage is pairwise comparison accuracy varies widely according to the number of reps for that entry. Here, alpha lattice, you can see the 20 plots across in columns, 7 plots in rows, and the blocking showing there for one replication for 140 genotypes and 14 blocks times 10 genotypes per block. Augmented can be very useful. There should be at least 3 different appropriate checks well distributed through the field. And checks are analyzed in an RCB design. They can also be used for positional adjustment of BLUPs to the similar to nearest neighbor analysis. So here, an example of 4 checks. Number of replications is 7. Sorry about the misprint there. Giving us 18 error degrees of freedom in this example, and mean square of 400 for error, and F-calcs derived from that error, and the probabilities, and then the BLUP estimates will take into account that degree of variability. Partial reps, and each environment contains 2 reps or even more of some entries, but only 1 rep of other entries. And software is available to handle that without a problem. Row column adjustments can be made using these column designations and the rep designation or row designations, and it adjusts for field variations beyond the blocking. And simplest approach is rows, columns, and rows by columns as covariates in ANOVA or linear mixed model, and it can be used in combination with any other any other design. I see I've got this repeated. Sorry, I thought I had that taken out. Now, final major portion, G by E is a very important element of our research. G by E is almost always larger than our pooled error, and multi-environment trials are essential to get accurate estimates. Now, environments, locations, and seasons have an unrepeatable and unpredictable effect on genotype means or treatment means, and therefore should be considered random. And all the interactions of genotype with those environmental factors should also be random. Therefore, G by E mean square becomes the appropriate denominator for testing the significance of genotypes. And that mean square becomes the appropriate error term for derived values, and the genotype by environment degrees of freedom are the appropriate degrees of freedom for those statistical tests. Analysis models and options, if they're properly constructed, should automatically take care of this. And here I won't go into detail, but the point is that to test the genotype effect independent of other sources, it has to be divided by the G by E term. Well, I'll rush through this one. And also here, let me skip to Amy and GGE. They're most useful for many genotypes and many environments. They're okay for student learning if you have at least 3 environments, but they're generally poorly interpreted, especially in practical application. And each has specific advantages, but you need to know what you're trying to get, what information you're trying to get to improve breeding program decisions. And defining mega environments is a large undertaking. You need 3 or more locations per potential mega environment tested in at least 5 seasons. I'll skip through this. We should be using multi-trait indices for selection, even simple ones. And I'll just say it, often there's too many technical reps being used. It's expensive and time consuming. So what's the optimal level? It depends on the number of biological samples, the level of analytical variability, and that is almost universally smaller than biological variability, and depends on the cost of consumables and staff time relative to other costs compared to the cost of the experiment. And decisions should be made guided by the standard error of the mean and standard error of the difference calculated from an analysis that includes duplicate sample data. All right, so are you using all available techniques to reduce experimental error? And there's several of them listed here. And I'll have further explanations and a more cleaned up PowerPoint at my website at pgibson@agimprove.africa. And you can contact me at that. And info from the presentation can be cited as personal communication from me today's date. So with that, I see my acknowledgements got messed up here, but in any case, I wish to thank the African Plant Science Symposium for inviting me and sponsoring this, and Macquarie University, the Department of Crop and Horticultural Sciences. So with that, I will stop sharing my screen and turn it over to my colleague, Alfred Ozamati.
All right. Okay. In the interest of time, I think I will try really summarize from what Prof. Gibson presented. First of all, allow me also to take this opportunity to thank the organizers for inviting us for this wonderful webinar. So I'm just going to share with you a little bit of my experience of integrating artificial intelligence, 3D physical modeling, and integration of integrating that into genomic tools for efficient crop breeding. So as already introduced, I think I'm not going to say much about what I am, but by and large, I'm a faculty staff at Macquarie University. And again, my research interest is in genomics, genetics, and genomics. So just a quick reminder here. Why is AI really considered to be critical in modern crop improvement? I'm sure these are probably familiar ideas, but just to remind us that we know phenotyping large breeding trials is not a simple task, and therefore we need efficient methods of quickly phenotyping, especially in large breeding trials. And the other thing we know is that there can be some subjectivity in data collection, especially when you use 2 or 3 different people. And so one way to minimize this subjectivity is to use AI-trained tools so that you minimize error. For example, think about collecting disease data. Then the other thing about using AI in crop improvement would be thinking of reducing cost of phenotyping. For example, think about nutritional traits. So if you do wet chemistry, for example, we know that it's not cheap assay for phenotyping, for example, large breeding trials. So that's also an area where AI will be handy. The other thing is just think about data standardization, because if 2 people collected data, say disease data with eyes, you're going to see differences in terms of how people assess, for example, the disease. So one way is to try and develop a model, use the AI model, and then that helps to standardize the data in space as well as in time. Maybe just a quick overview of the workflow, especially when one is interested in using AI for phenotyping. So basically, this is image-based kind of models that are trained. So it all starts with collecting images that reflect the traits that we're interested in breeding program. Then these images are cleaned. They're labeled so that they have identification. Then basically, then once you have your images, the next thing is you also have to have the conventional way that you generate the data. So then eventually from there, using your AI-generated data and then the ground truth data that you collect, you get into what we call the model training state. So where you split this data into what we call the training set and the validation set. And then once you get to a stage where your model can well predict the trait of interest, the next thing is you get to a phase which is more of what we call the testing stage. So that's where the translation bit then happens. But to bring this to context, again, I just want to share my experience from work that we have been doing in collaboration with UC Davis, basically trying to generate data using AI and also looking at our traditional method of generating data and see whether these really answer the relevant biological questions that a plant breeder will be interested in light of the experimental design that Prof. Gibson just discussed. So for this particular experiment, we used 50 diverse cowpea lines that represent some bit of global diversity. And so we established this trial at 2 sites. And then we planted it using alpha lattice with 2 replication. And so the data that we generated, both using the AI and ground truth, were plant heights, days to flowering, days to maturity, and yield. So these are just the layouts. So the team, the other side is basically collecting the conventional data. And then the other side is basically the mosaic pattern of data that is generated from AI. So how do we do this? This is a workflow of basically how we really generated these images. So through this project, of course, we basically use 2 data collection modes using AI. So one is drone generated data images, and then there is that data that is also generated from rover. But for today, I'll just talk about the drone generated images. So basically what the team does in terms of the workflow, or what we do in terms of the workflow is that, so we basically load a mission. So the mission means we load the reference field where we want to really fly this drone. And then the project itself has developed a particular application that helps to process those images, which we call field image AR. And then once these images we generate from the field, we collect those images from the field, then we plug it in the software. Then the software helps us to generate what we call orthomosaic field layout. So that's based on the images. But then also we have, of course, the images are then processed. You can see, for example, here, for example, the image will be processed into something that a trait that we already know. And then we generate a table like this, which is the image generated kind of reflection of the traits that we're interested in. So that's what it is in terms of the workflow. Just to give you a quick feel of how we have utilized this data in the breeding program to make decisions. Again, having an AI tool is a good thing, but once you don't get excited about, for example, if the students here, sometimes there's an aspect of getting excited about technologies. So one thing one will do is to quickly look at the repeatability of this data, which presents the biological relevance of the data that the breeding program is interested in. So in this case, I have examples of a few traits. So the plant count. So here we have days to flower, 50% flowering, days to maturity, 50% maturity, the pod number, and then the seed weight. So basically looking at the heritabilities of this, you could see potentially some of these traits actually quite have good repeatability from this AI-generated data. But of course, we have plant height, which basically has sort of low repeatability. And for cowpea, for example, this is understandable given the height above ground not being that much, and also because of the way sometimes some varieties grow spreading and so on. But then further, the other question is, so if you have the AI-generated data, and then you have what the breeders are traditionally used to collecting, and then one thing you want to see is whether that data that AI has generated rhymes to what the breeders basically measure in their traditional way of measuring phenotype. So again, the example we looked here is for plant height. I did this intentionally just to show that the biological relevance actually has a tie to eventually the relationship that you'll see between an AI-generated data and the phenotype. So for plant height, remember before we showed the repeatability was low. That means the repeatability from plot to plot was. So even again, when you try to now correlate the data that is coming from AI-generated images with what the breeders themselves measured for plant height, you see the relationship is low. So that has to do with the initial data quality, isn't it? So that ties to what Prof mentioned, that if you don't do a good job at the level of designing trials, even when you have the AI tools, you're not going to perform a miracle. So the other thing I wanted to share with you is beyond this, of course, let's assume we're comfortable with AI-generated data. So then the next thing is in terms of translating this into breeding. I want to have more efficient way of using this data for breeding, isn't it? So we could integrate into genomics into the actual data usage. So that's for the downstream application. So again here, this is data that was, basically this analysis was done by our partners from UC Davis. So they basically generated a bunch of traits. So what you see on this side are basically AI-generated data from images, and then this is what typically breeders will go and score, or what the technicians will collect from the field. So the thing I wanted to show here is, of course, this was done with about 168 photoperiod sensitive cowpea magic lines, which represent diversity. Data collected manual and using AI sensing tools. And then, of course, there is a whole SNP chair isolate SNP array that these 168 were genotyped to it. So basically what I wanted to show here is, for example, let's focus on, I'll be interested in, for example, let's look at this average flower count here. That is from the AI-generated data. I hope you can see my cursor. And then you look at the genome prediction accuracies. Compare that with this, again, this particular data, which was the predictions coming from manual generated. You can see there is some bit of equivalence in terms of prediction accuracies. Although the manual or conventional generated data has a higher prediction accuracy. So this shows that for this particular trait, the AI is actually picking something similar to what also manual can collect. So then if that's the case, eventually breeder can actually potentially use AI data, which has got the advantages that I mentioned on to do genome prediction and shorten breeding cycle. The other example I just wanted to also share is using that same AI data for trait mapping. And we know that one of the easiest ways to shorten breeding cycle is to use markers. But to do trait mapping, of course, you need phenotypes. You need the phenotyping, again, I said, if you use conventional methods, there's some of that the advantages. And if you use AI, I think I already mentioned at the start, some of the advantages from AI generated. So this particular tutorial mapping here just shows a comparison between data that is again generated from AI for flower count. You can see more or less the QTL on chromosome 9 is detected using both methods. And in terms of the signal of the QTL, it's basically about the same. So meaning potentially the data sets that is generated from AI potentially calibrates or relates well with the manual generated data. A few thoughts from me based on the involvement in this project. Well, I think as we think of moving forward with having new innovations integrated into breeding, AI is going to be critical for us to increase breeding efficiency, for example. But let's stay warned based on the conversations that or the views that Prof. Gibson just presented, that don't forget the classical experimental designs to ensure that you have the right quality data. And that's the only way you can calibrate the AI-generated data well and make use of it in real breeding. So that said, let me acknowledge the people that I work with, UC Davis. That's really the one that is strongly supporting work in terms of the funding. But of course, there's also a couple of other people who are part of this network. With that, I think I will end my presentation. I will be happy to take